What you should realize by now is that we are not alone, and at any given time we have trillions of bacterial cells living in us or on us. So what we'll talk about now are the basics of prokaryotic cells and how they differ from eukaryotic cells. Originally, all living things were put into 5 kingdoms, and Monera was the category for prokaryotic cells where bacteria were found. You should remember that the term Monera, you'll see it in text, and you might see it on a standardized test. I hope not, but it is possible, so you need to remember it. In the 90s, things were changed into 3 domains, archaea, bacteria, and eukaryota, and on the left you can see all of the characteristics or the divisions starting with life down to species. And this is how it is currently organized. So in the 6 kingdoms are the prokaryotes, which are the archaeobacteria and the eubacteria. And then the eukaryotes hold everything else, protista, fungi, plantae, and animalia. So here's another view of this. It's more of an evolutionary picture. You can see that the eubacteria and the archaeobacteria came first, and then the protista. And from the protista came plants, animal, and fungus. And everything is a eukaryotic cell except for the eubacteria and the archaeobacteria. One of the main differences between eubacteria and archaeobacteria is the construction of the cell wall. In eubacteria, it contains a sugar called peptidoglycan, and it's not found in archaeobacteria, and that's one of the reasons why they split them up. So an example of eubacteria might be bacteria that we come in contact with every day, and then cyanobacteria, which makes up pond scum, basically. We'll talk a little bit more about that. Archaeobacteria are the methanogens. They live in your gut, and they cause the biological accidents that your peers like to point out. Halophiles, halo is halogens, which is salt. They're salt lovers. And thermoacidophiles, right? Thermo, they like heat. Acid, they like acid. So they live in rather harsh conditions. We usually don't come in contact with archaeobacteria as often as we come in contact with eubacteria. This is a diagram of a simple bacterial cell. It has a capsule which adds to its structure, a cell wall. Cytoplasmic membrane is another name for cell membrane. It has ribosomes that make protein. Pili helps with, they're sticky and they help bacteria hang on to each other so that they can exchange genetic information when they reproduce. It does not have a nuclear membrane. It does have DNA, and it tends to be in a circular fashion. The flagella is that long tail, and it helps with movement. And that's about it. Very, very simple cell, one of the first cells. And it's considered to be a prokaryotic cell because it has no nuclear membrane and no membrane-bound organelles, like mitochondria and chloroplasts and those kinds of things. In comparison, these are eukaryotic cells, the animal cell and the plant cell, and they both have a nuclear membrane, they have mitochondria and or chloroplasts, they have vacuoles, centrioles, and Golgi apparatus, endoplasmic reticulum, very complex as compared to the bacterial cell. So again, they have membrane-bound organelles, and prokaryotic cells do not. Take a look again at a prokaryotic cell. No nucleus, no membrane-bound organelles. You get a sense of their size from this picture. This is a picture of the tip of a needle, a sewing needle. and you can see that they're very, very small. This was taken from an electron microscope. We'll have a hard time seeing them in our microscopes because they're so very small. Prokaryotes can be identified by their shape. Spirilla, obviously spiral. Bacillus are rod-shaped. Look at the L's in the bacillus. That'll give you the clue that it's rod-shaped. And then cosi. Look at the word. Almost everything is circular. See the O's and the C's, so that will give you a clue as to that they are circular. A disease that's caused by spirilla is Lyme disease. Anthrax is bacillus, and strep throat is caused by a cosi. Gram staining is another way to identify bacteria. It's just a series of stains and washes. If you take microbiology, you'll go through this. If you look at the diagram on the right, you can see the difference in the construction of the cell wall. Gram-negative has a very small peptidoglycan layer, and the gram-positive has a very large one. And so gram-positive tend to be much more rigid of a bacteria. So they can look at the stains and see what color they are and identify them. The other example of eubacteria is cyanobacteria. It has a lot of different names. Blue-green bacteria, blue-green algae. It's most often now called as cyanobacteria. I don't know why they keep changing the name, just to make sure you're alert, but it's basically just pond scum. You might sometimes find it in your swimming pool, but if you have made a trip to Bee Meadow Pond, and if things aren't looking very good, you might actually see it here. This was taken at a lake near my home, and you can see the pond scum. It's kind of dangerous. Some cyanobacteria give off a toxin. Not all, but some do. And we tend not to get too hung up on it because this pond scum's pretty smelly and stinky, and we don't really like to swim in it. But you should avoid it because if you do swim in it,